Our next presenter is Konrad Krawczyk, talking about NaturalAntibody.
Thank you. We're actually running ahead of schedule. That's great. Hope I did not spoil that. All right, thank you very much for having me. So I'm going to talk about federation of antibody data for immunoinformatics and biologic discovery. So essentially, like internally, we just talk about playing bingo with antibody data and modeling tools. And why do we do that? So just a bit of context for that, because we are very much a computational antibody company and lab. So we want to use our data, modeling, and expertise to just apply it to internal discovery projects, be it in academia or in the industry. So for that, of course, we needed a bit of a plan how to actually not only have our antibody expertise, but also to develop a set of tools that we can use for it. So the very first bit of it was, of course, you cannot see the pointer, was of course getting the data, because this is the basic resource for any modeling. And this meant identifying or getting as close as you can get to all the antibody data in the public domain that are out there, but also just to develop some of the experimental data ourselves. This is all used to develop modeling tools. And they are supposed to cover a very broad spectrum of what you can actually apply to antibodies. And this is, I cannot stress this enough, we do want to perform all the modeling ourselves, just to consolidate this expertise internally. And then of course, and this is the point about the demo here, just to make our lives a bit easier, but also just to give the tools to some people in either academia or in the industry, we do put those into a platform that is supposed to make access to this data and to the models slightly easier. So of course, first step in this is the data. And there are many excellent databases out there focusing on antibodies. And what we just had an issue with was that we wanted to just get as close as we can get to just getting everything that there is in the public domain. That meant mapping the landscape of the sources or the different data types. And of course, some sources are easier to get than others, some you can automate, some you need to perform manual curation, or a bit of both. You need to find the common denominator. And for us, this was just the variable region sequence. And what you do with that, of course, there is data that you can just get, the sequences, you can just divide them pretty nicely. You can get the metadata. It's quite hard to actually systematize that, but it's not beyond impossible. And what you do need to do at this stage as well is actually add some more layers. Yeah, so the embedding and indexing layer to make it possible to firstly explore all this data, but also to make it possible then to create data sets for modeling. And then for retrievals and actually further to just chop up this data, you need to apply some natural language processing to, for instance, NER to identify targets, and then systematization, because not all the gene and protein names are reported in a very systematic way. And of course, because this is the AIRR conference, I just wanted to talk a bit about just the NGS or AIRR component in that. So back in the day, when we were collecting the NGS data sets, we, let's say by manual means in 2018, we identified around 50 bioprojects that we put into a database. These days at NaturalAntibody, we just did automatically. So we went through all of the bioprojects that we could find. So it was around 500,000. And now we just identified around more than 200 bioprojects. So here I divided between those that we are very confident about and 24 that are slightly less of a good quality. And most of them are Illumina, and they're around 25 billion row reads, 27 terabytes, which makes it really fun to train models using that. And actually, most of them are from the last 2 years. Yeah, so this is something that's good that it's picking up pace. So then finally, when you get all this data in one place, you can, before you even get to modeling, you can put it into a search engine. Yeah, so basically, with all the data that is in there, that I just briefly flew through, everything just can burn down into just two search options. Yeah, one just the sequence search. Yeah, if you wanted to see if there's anything out there that is similar to your CTR3 or a variable region sequence, or going the other way, just the keyword search to see if someone has ever been developing an antibody that was associated with a disease target or something else. And of course, we are a company, but some of the bits that we develop do percolate into public domain. So I do encourage you to, for instance, just check out the INDI database, which is just the such integrated extract, but just for nanobodies. Then of course we get to the modeling bit. So because this is actually our spiel, and we do wanted to just when all the new data comes in, to create frameworks that do train the machine learning models or statistical models in a very consistent manner. Yeah, because we don't want our models to be out of date anytime soon. But then again, you do have this repository of models in your backend, and you still want to be able to use them. Yeah, we want to be able to use them internally in a hassle-free way, but also just to give them to other people in the community. So here, of course, there could be a myriad of views for different modeling tools. We try to burn it down to just two for now. First one is just the single molecule view, which is supposed to just give it a sequence, and it's supposed to give you the machine learning annotations associated with different residues. Yeah, like positional mutability, developability contributions, and so on. Then, of course, the other bit, which is just the batch annotation, and this is when you have a set of sequences and you want to just filter them using different machine learning methods. And of course, here, we also percolate in the public domain. So I do encourage you to look at AppDiver, published recently in Bioinformatics. And of course, because this is a lightning demo, so the very point of it is just to show that I'm here. If you're interested, just come and talk to me. And by interested, I mean we also work quite a lot with academic labs. So if you have a nice use case, just please come and talk to me, because essentially we are in the business of just making the data and the models quite accessible. And of course, thank you for the organizers again, and thank everyone at my team for actually doing that. Thank you.
Questions for Konrad? I have a quick question, if you don't mind. You mentioned that you can do a keyword-based search of sequences. How good is that? And do you get some sort of confidence on the association between the keywords and the sequences?
By keyword, I mean essentially in text. Yeah, so I just this is a difficult question, because normally what we do, we just benchmark it using documents that we already know. Yeah, but obviously if there are some benchmarks out there as well that we just do that are normally used for text retrievals, Elasticsearch and so on. So we were using them. It's slightly more difficult with synonyms, because this is actually one of the biggest drivers for a keyword search. But here, and the sources that actually do curate the synonyms for genes and proteins, they actually do not agree to a huge extent. Yeah, so actually we are right now I'm not trying to give it in the backend as was previously, but actually just show this is the list of synonyms that we actually identified. Are you sure you want to start using that?
Great, thank you. Any other questions for Konrad? All right, thanks very much, Konrad.
Thank you.